In this exercise, you will determine the A, B, O blood types of unknown blood samples. The blood used in this experiment is artificial and free of any pathogens, so it is safe to use. However, in this lab, we will follow precautions used for working with real blood, as we are simulating real-life conditions. According to these precautions, you will wear gloves at all times when handling the blood. Disinfect the benchtop and any other surfaces or equipment that might have been exposed to the blood, both before and after performing the procedures. Dispose of all materials that have been exposed to the blood in the biohazard waste container. Each group will test 2 unknown blood samples. For each sample, you will use a separate plastic blood typing tray. In each well on the blood typing tray, place a drop of the corresponding anti-serum. Anti-A serum in the well labeled A, anti-B serum in the well labeled B, and anti-RH serum in the well labeled RH. Next, place a drop of the unknown sample in each well. Be careful not to touch the tip of the dropper bottle to the well. This can contaminate the bottle and transfer the antibodies to the other wells. Use a separate toothpick to mix the contents of each well. Discard each toothpick in the waste cup after you use it. It is very important to use a separate toothpick for each sample so as not to transfer the antibodies to the other wells. Incubate the slide for several minutes. Periodically, rock the slide gently. Be careful that each sample stays in its corresponding well and that the samples do not mix. After several minutes, observe the samples for clumping, which indicates hemagglutination. When you are finished observing the results, discard the toothpicks into the biohazard waste container. Immediately clean the plastic tray with soap and water. Otherwise, the blood may dry onto the tray. Remember, this is artificial blood and anti-sera, so we can reuse the trays. When you are finished, be sure to wipe your work area with disinfectant. During the lab, follow the directions in LabTutor and ask your lab instructor if you have any questions.